Hi, I'm Karina Cannell and I'm an undergraduate researcher in the Rose Lab here at UCI. Today I'm excited to share with you a little bit about our research where we are exploring the biological mechanisms of lithium on Healthspan in Drosophila melanogaster. What if you could take a pill that would make you live forever? But what if it had a cost of poor health? Would it be worth the risk? This brings the focus on health span rather than lifespan. But what is health span? It is the length of time that an individual is healthy, not just alive. So how can we improve health span? We believe that lithium is a potential candidate. Recent studies have shown that lithium extends health and lifespan in different animal models. It is also commonly used as a treatment for bipolar disorder and depression in humans. But why is lithium so good at extending health span? And why are we qualified to answer this question? Here in the Rose Lab, we maintain large outbred populations that have long-standing adaptation and massive replication. So some of the populations that we are testing lithium on are our A types and C types. Our A type or accelerated aging population have been selected for earlier development, and as a consequence, they have a shorter lifespan. And this is in contrast to our C or control types, which have later development and a longer lifespan. Previous studies have shown in our lab that the lithium has effects of an increased lifespan in control type flies, but that it negatively impacts their reproductive success. So our next step for our UROP project is to look at lithium's effects on A-type flies. And we're going to do this by conducting both a mating and fecundity assay, as well as looking into transcriptomics in the future. So for our mating assay, we give a male lithium and then see whether that increases or decreases the likelihood of being chosen for mating by a female. For our fecundity assay, we examine how lithium affects the number of eggs laid by a female. So we expose a female to lithium and see how many eggs she lays, and we compare that to our control fly. For our future direction, we hope to look at transcriptomics, which is the analysis of the RNA transcriptome, and it provides insights into gene expression. And by looking at flies exposed to lithium with transcriptomics, we can see how lithium changes the flies at a genetic level, and also see whether they have an effect on the gene expression and what its mechanisms are that help increase health span. What I'm most excited about in this project is that it's translational to human health research. We're not just looking at the trade-offs of fly populations with different genetic backgrounds, but we're also looking at the mechanisms behind such a commonly used drug in the medical field. We'd like to thank the support from our wonderful mentors and the funding from UCI Cal IT too. Thank you for listening.